Great. Thanks for joining us, everybody, for our next installment of our mental health seminar series, Research Exchange and Conversations on Metabolism. I'm really excited about this intersection of things that maybe aren't traditionally considered related, but of course are. We're going to learn about that today. So we have two wonderful speakers with us. We're going to start with Dr. Craig Ferris. Dr. Ferris is an interdisciplinary professor with joint appointments in the College of Science and Bouvé, as well as director of the Center for Translational Neuroimaging. His academic research focuses on developmental behavioral neuroscience and disease progression in psychiatric and neurological disorders in the hope of improving psychosocial and psychopharmacological intervention strategies. And Craig, if you can take it away.
Thanks, Vance, and good morning, everyone. It's always nice to be invited to talk on a topic that you essentially that you don't, it hasn't been your main focus of research, but everybody seems to dabble with this peripherally that does anything in the area of mental illness. And it really is this quest for biomarkers, as I entitle this, the clues to mental illness, complex disease, as we all know. When we talk about schizophrenia and depression and autism, not easily modeled on my side with animals. And so it's almost like the last great unknown in terms of disease in humans. And so it's approached, and I brought this up because, this goes back 20 years. And I've been in this business for 43. So there was a lot of excitement around this. And this was something that came out, the genome era and mental illness. And so this came out by Francis Collins over 20 years ago. And so this is from the National Association of Mental Illness. And there was so much excitement about if we can just break the code, if we can sequence those 3 million DNA letters and come up with the human genetic blueprint, we have a way of approaching mental illness. And right there, that last line is the dawning of the genome era in medicine. And what was conceived of at that time was that if we could identify all these genes and then identify their mutations, then we would have a handle on, well, okay, that gene is mutated. We could develop diagnostic tests. When you look at this little schematic that came from Collins, and if you have the diagnostics, today that, the diagnostics are used during pregnancy and they're used afterwards to kind of predict diseases and, give you a better understanding of your options, your choices. And so in terms of prevention and pharmacogenomics, identifying the genes and gene therapy, we have marvelous technologies now for correcting genes that are mutated. And then understanding the biology of the disease and targeted drug therapy. So this was all laid out almost 20 years ago. And it really hasn't met expectation. I think I can honestly say that as it relates to mental illness. Yes, there are many, many genes with mutations and many genes that are risk factors. So then begins a different era. And this is the era of the, as I call it, the omics. And it began with the genome and identifying DNA. Then it moved on to RNA and the transcriptome. And from there it went to proteomics and making proteins. And I know in some of my studies that I do that I'm looking for metabolites of brain injury, okay, I focus on proteins. And what proteins are appearing as biomarkers that tell me that there's been a mild head injury. Okay, that's good, but what seems to be getting more attention and seems to be almost the better way to approach this larger picture of mental illness is metabolomics, where you're looking at readouts, again, biomarkers in carbohydrate metabolism, the nucleotides, the amino acids, and I highlighted lipids down there in the lipidome, because that's becoming even more prevalent of all of these. That seems to be the direction. And all of these metabolomics are giving you biomarkers of breakdowns in what would be normal metabolism and what's going on, okay? And hopefully providing some understanding of the phenotype over there. I say the lipidomics seems to be getting more traction in all of this. And from my understanding of it, it's really a chemistry issue. The chemistry issue is this: you get hit on the head. This is my work we've been doing. You get hit on the head, you're waiting for proteins to cross the blood-brain barrier so you can get a readout. Not easy, okay? But if you're looking for lipids to cross the membrane, that's much easier. So I think a lot of this going into lipidomics as a readout, a biomarker of what's happening in the brain, is getting a lot more traction. And I think that's something that we certainly should consider as here. So I did I went to PubMed and I said, so what's all of this metabolomics and what are we doing with it in mental illness? And so this is just a quick search I did over a decade from 2012 to 22. With metabolomics and mental illness, I popped up 1,300 publications. Alzheimer's stands as number one. Metabolomics and mental illness, and this really relates to dementia. And so there's all of those references. Next on the hit list was depression with its 224, but surprisingly, the next was gut. Okay, this movement in the gut-brain interaction and our microbiota. After that came schizophrenia, basically one and one here. Then autism, then drug addiction, and anxiety disorder. So this is kind of how it lays out this connection between metabolomics and mental illness, and just the kind of work and the kind of studies that have been going as to the kind of studies that people are doing. All right. Actually, before I leave this, I'd like to highlight gut and drug addiction, because that's what I'm going to spend a little bit of time talking to you about that. I'll mention it again. So here's, so I said, there's all these papers out there. So what are they about? There's a lot of stuff on autism spectrum disorder. But look at that. This is gut-brain axis. These are the questions that are being, or studies that are being done as it relates to this, the gut brain and the gut microbiota. So there it is, altered gut microbiota profiles associated with abnormal metabolism activity and autism spectrum disorder. And all of these are very, very recent. They're all 220, actually, 221s. So they've been using the gut-brain axis to study post-traumatic stress disorder. They've been using it for autism spectrum, major depression. And down on the bottom there, you have anorexia nervosa. So, and I'm certain, this is available to everybody. I'm not only taping this, but can people see this? Vance? Yes? Okay, good. Okay. So you can save those references. And so there's just, again, this is kind of a schematic of all of the things the microbiome has been associated with respect to and mental illness, and the papers are just pouring out. And so the microbiome, the gut actually regulates and affects dopamine and serotonin levels, actually creating precursors to them. You always think about that gut feeling. It's used a lot. The enteric nervous system has more neurons than our brain does. All right. And so here you have this incredible enteric nervous system associated with the physiology and the coordination of physiology in the gut. All right. And it speaks to the brain, and it speaks to the brain through the vagus nerve. And there's a lot of work that's being going on right now looking at vagal activity and the gut microorganisms. And a lot of that is manipulating directly activity in the amygdala, the hippocampus, prefrontal cortex, the usual suspects one considers when talking about mental illness and behavior. So that's kind of an interesting slide. And again, I'm just bringing your attention to the fact that there's so much work being done on that. So this is how we're dabbling in it. And of course, like many things that we do in our lab, it's really not by design, it's just by, serendipity. And the serendipity here is that Charles River Laboratories got in touch with us about a year ago and said, listen, we've got these germ-free mice. Okay, no one wants to buy them. I guess no, so they actually gave them to us and said, do something with them. And so so I have a student, Sade Aria, and Sade Aria studies oxycodone addiction. She studies this in rats, and she does all of the imaging, and she's able to she's able to use functional and structural imaging, magnetic resonance imaging, to follow what is called neuroadaptation. What happens to the brain when you're exposed to drugs, and how does it change with chronic exposure and addiction? So anyway, she looked at this model and she said, well, I wonder if these mice are responsive to oxycodone, hence the title. They come in with no bugs, as I call them, bacteria. Or are they less sensitive to opioid addiction? And so that's what I'm going to give you just a little brief introduction to her work. She's trying to finish this up right now, and I'm sorry I don't have all of the imaging that goes along with this, which is also pretty stunning. But looking at this cartoon, let me just quickly bring you up here. The left-hand corner. So in come these mice from Charles River. They're the wild type, have the complete microbiota. The germ-free don't have anything. All right. So Sade says, can these animals get addicted? So what she does, and I don't know whether you're familiar with this, just so-called condition-place preference. It's very, very simple. Let's say you have two chambers, all right? And the study starts off by taking all of your different subjects and saying, which chamber would you like to hang out in? They're given a choice at the beginning of this study. And so they have the preferred chamber and the one that they would rather not go in. So the way she set up the study, she took all of the germ-free, she took all of the animals that were going to get oxycodone, and she injected them with oxycodone in the room they would rather not be in. And she injected the other ones with saline in the room they would rather be in. So you're really hedging your bet against this penchant for drug abuse. So let me get back there 1 second. So she comes back, she does this drug association chamber stuff, and then she does the test. And what she discovered was everything in blue here are saline treated animals going to the room they prefer. Everything in black here are oxycodone injected animals going to the room they would rather not go to. And what she found is, yes, there was a significant difference between the germ-free and the wild type, okay, in the saline here, but they were less sensitive. Okay, so they weren't as responsive to the oxycodone, and she's doing all of the resting state functional connectivity and how the brain is organized. And hopefully we were hoping to have that data to present here. But I'm excited to see, because what happens next in these studies that's so neat is something called conventionalization. If you take these animals, the germ-free and the wild types, and you put them together in this little, they just allow them to cohabitate in the mouse cages, they will transfer the bugs. Okay, and so 30 days later after this cohabitation, all right, and we've already done all of the fecal analysis. So we've already had the sample sent out, and we've got a full display of what the complete bacteria, look like. And so these animals come back and they have the same bacteria. All right, now you repeat the same test. And I'm not going to, belabor this anymore. Same thing. And now when you put them in the condition place preference, there's no difference between them. All right, now they have full sensitivity to opioid addiction. Interesting study. We're involved with a company right now. It's called Holobiome, which you may be familiar with. It has an association with Northeastern, certainly with its faculty. And we're going to, pursue this. And we're pursuing this from what bacteria are responsible for this. And so this is one exciting way to do this. In the larger picture, so this is our drug picture. Here's the, when you dig up the literature, how do you approach these kind of studies? So you think about how would we approach these on our campus here? This is a roadmap for the, the microbiota studies. And so here you've got your control and your patient group. So this may be autism, this may be schizophrenia, this may be depression. And you literally, you recruit the groups and collect their feces. Okay, and the stools get sent out for analysis. They get sent out for metabolomics as well as fecal microbiota. So you're basically finding biomarkers that would describe this condition. Not easy. Actually, what's even more difficult is the next step. So now I have this population. Can I somehow relate this population to an animal model? All right, can I start to compare these? And this is where it gets difficult because the animal models for mental illness are very, very difficult. And it would be wonderful if we had, cynomolgus monkeys and marmoset monkeys and non-human primates on our campus, and to bring all of these translations closer to the human condition, but we don't. Okay, so we deal with the models that we have and hope that they can inform us. All right, and they do. There's a lot of information that we can get out of our models. And basically, this is what we've done. Okay, we find our mice. Okay, they have a problem with, not a problem, but they have changes in addictive behavior. With and without the microbiome, we do all our, our testing, which we're doing. So what do you do now that you know this information? We're taking the approach with a company that we're going to see if we can identify the bacteria that are involved in basically this kind of reward behavior. And this would extend into not only addiction, but this could extend into numerous other things. For instance, eating disorders. But anyway, so as uncomfortable as it might seem, part of the solution is fecal transplantations. You're taking healthy people, and they're doing this, and there are companies that do this. You take healthy people and you isolate their fecal microbiota and you transfer it to the unhealthy people. And these studies are ongoing. There's also one that deals with just, how, this is not sexy, but it's approaching lifestyle changes. What you eat, okay? Can we influence these disease conditions by, prebiotics, probiotics, food, exercise, lifestyle changes? So I think it's an actually interesting area of study. My lab is going to continue to do work in this area if anybody might be interested. We're also doing a lot of work on mild head injury, which I think we're going to be using the lipidomics to understand those biomarkers and by then, okay, some of the mechanisms underlying the head injury. So this is where we are right now. And Hazel, I'm going to pick, I don't have anything after this one, so I'm escaping here, and I will stop sharing. If anybody's got any questions, go ahead. It feels new to us. And I invite anybody to come and play if they're so interested.
Thanks, Craig. Yeah, are there any quick questions from the audience right now? Otherwise, we will have time for more after Hazel.
Maybe we should bundle at the end.
We can do that too. Oh.
So sorry, I have a quick question. So my name is Motaz Al-Salman. I work with Dr. Love. So do you look at the control group, what they eat? Do you investigate their diet when you recruit them, or you just randomly select them in that study? Thank you very much.
The data, the diet is simply the diet that's provided by our animal medicine. Okay, so they don't have unique diets. When they come in, they're germ-free. If you did nothing else with them and tried to isolate them, they would eventually get the germs. They would get the germs from being handled and so forth and so on. So the diet, no, but the diet is a really interesting point. This diet is a chow that's used by everybody. Okay, this is standard chow used by animal medicine, and I cook everywhere. But that's an interesting question, and one would start by approaching it. Can you approach this by changing the diet?
Let's move on to our second speaker, and we can do follow-up questions for both at the end, just to make sure we have time to get through everything. So thanks again, Craig, for your lead-in presentation. It was really interesting. Next up, we're going to have Dr. Hazel Siv, the Dean of the College of Science and Professor of Biology here at Northeastern. Dr. Siv's groundbreaking research focuses on neurodevelopmental disorders as well as fundamental processes underlying brain and craniofacial development. She is a distinguished educator, a MacVicar faculty fellow, MIT's highest undergraduate teaching accolade, and twice a winner of the MIT School of Science Teaching Award. Hazel, if you'd like to, I think you should be allowed to share as well.
Yep.
There.
Am I sharing?
Looks good.
Thumbs up. Okay. So nice to see everybody. Thanks, Vance, for that kind introduction. This is really brilliant to be able to have this conversation. And Craig, thank you. You've told me about this work, but wow, what a crazy important finding you've made with regard to the question of microbiome and opioid addiction. It's really my pleasure and honor to be a member of the faculty here, to be dean as well. I'm delighted to help lead the university here into the future, but I'm also delighted to be a faculty member here in biology and to contribute to the future of science here. One of the things that my research group studies is neurodevelopmental disorders. We also study craniofacial development, but I won't talk about that at all. And I want to do something that kind of takes what Craig gave you and makes it more pointed. So I'm going to talk about a particular neurodevelopmental and mental health disorder, and then I'm going to talk about the contributions of metabolism to this disorder. So I think I'm going to, is it okay like this if I keep my little slides on the side, or it's okay? Because then you can kind of see where I'm going, and I think maybe we'll be more on the same page here. Like Craig, and this is fabulous, we have also done an analysis of the genes associated and biomarkers associated with mental health disorders, as has, a huge community of people who are doing genome-wide association studies. But it's been particularly interesting to us to pull out those genes that encode enzymes or enzyme modulators, not synaptic components per se, but more biochemical modulators of cellular function. And if you do that, here's for example, that Monica Saxena pulled out for bipolar disorder. You can see that about 20% of the GWAS hits, the genes associated with this particular bipolar disorder, are involved in metabolic processes. So they are either metabolic enzyme encoders or modulators of metabolic enzymes. And this is very interesting because at least some members of this class of associated proteins or readout proteins are fairly druggable. Some of them are not so druggable, but some are druggable. And the question of the metabolic processes that might be associated with mental health disorders really has been on our minds. So it's been on the minds of affected people as well. And there is hope that diet can cure mental illness. Generally, there's a huge, and there's an industry here. I've shown you a bit on this slide. There is not so much substantiation of this notion, but except for omega-3 fatty acids, which may be efficacious in some cases. But, this is a very popular way of thinking about things at a more human accessible level. In fact, what I'll discuss is that maybe this is not so far-fetched, but it's not sort of random off-the-shelf stuff that is going to make a difference. It's more biochemically identified, substantiated targets that could then be tweaked with some chemical modulation into things that look like dietary supplements, but are very much targeted to the particular disorder and, into perhaps this popular notion of personalized medicine, which, one hopes is not so personalized that you have to make a therapeutic for each affected tiny landscape, but can be more broadly useful across larger groups of people. So, okay, so hang on, here we go. Let's move on here. All right. We started becoming interested in an experimental way in metabolic connections with mental health disorders by studying 16p11.2 deletion syndrome. This is a very severe copy number variant disorder. It's present in about 1 in 4,000 births. It is a de novo deletion, and there is 100% penetrance based on a study in Iceland that if you are deleted a region on chromosome 16 that encodes at least 25 genes, you are affected in ways that are variable but fall into a common bucket. The region is associated with autism, accounting for about 1% of all cases, epilepsy, hyperactivity, intellectual disability, hypertonia, and body size disorders. There are 25 genes associated. 9 of them, interestingly, encode enzymes or enzyme modulators. And it's clear that there is no single gene that is causative of 16p symptomatology, leading to the notion that there is some kind of combinatorial control among the set of genes. We have used the zebrafish as a model to understand something about this and other neurodevelopmental disorders. I don't have a slide plugging zebrafish. So let me just tell you, firstly, that we have built a zebrafish facility on campus in the basement of MUGAR, right near one of Craig's research labs. So that is a very nice connection there. It is for collaboration across the campus. We also have a very nice frog facility. So if you're interested in working with zebra, you just drop me a note and we'd be thrilled to collaborate with you on your particular project. But the reason that we chose zebrafish long ago is that they are very accessible during developmental stages in ways that other animals are not. You can get the embryos right from the fertilization onwards. They're transparent. You can get hundreds and hundreds of them. They're small, so you can do multi-well assays and get extremely significant numbers of animals involved. They are much cheaper to look after than mice. And, you know, they have neurons and brains that are pretty much the same as humans. Not exactly, but, you know, very related. So we have for many years now used zebrafish as an entry point into understanding genes and gene function associated with human disorders. In the case of the 16p deletion syndrome, we made a map of pairwise homolog gene interactions based on the notion that there is no single gene associated with 16p deletion syndrome symptomatology. But we made a pairwise map of the gene interactions, and here it is. And it was, and we found that there were a number of genes that had quite a large number of interactions. And the font size here tells you how many gene interactions each of the ones Jasmine McCammon pulled out. One of the ones that really caught our eye is FAM57B, a gene that we had never heard of, and probably you have never heard of either, but there you are, one is guided by the science. And so we were guided by the science towards FAM57B. There was a single paper on this in the literature, which suggested that this was a ceramide, that this encoded a ceramide synthase. And as you can see, it interacts with a lot of other genes. And what's fascinating about this list of interacting genes from the 16p cohort is that all of the enzymes and enzyme modulators that are in this interval came out as being part of this interactome. So this in itself is a rich kind of a landscape. But we decided to look at the contribution of FAM57B in more detail. In the meantime, FAM57B has been identified as a single gene, autism risk gene, as well as part of this 16p cohort where there is multi-symptomatology associated. All right, what are ceramides, you might ask? So ceramides that FAM57B was suggested to regulate are a class of sphingolipids, which means that there's a sphingosine moiety, and then there's a long chain ceramide part, and that gives you a sphingolipid. And these ceramides do lots of things. They are involved in building the plasma membrane and all cellular membranes, and they're also regulators of cell death, of cell cycle, cell proliferation, inflammation, and so on. It's a very important class of lipids. Danielle Tomasello, who was a very talented postdoc in the lab, went on to pick up the project that some very talented investigators before her in my group had been studying. And what I'm going to tell you in the next few minutes is that she, together with collaborators, went on to link FAM57B function to affected 16p deletion neurons from people and zebrafish. And she made the connections through lipid metabolism, synapses, brain activity, and behavior. This is kind of a summary of where I'm going. So I'm giving it to you at the outset that we will travel together from the brain into lipid membranes, into protein localization, brain activity, and behavior. And let me just say, I'd be thrilled. I think it's nice to bundle questions at the end, but if there's something that is totally mysterious to you and makes no sense, please do stop me, because it probably doesn't make sense to others as well, and you would be doing everyone a favor. So don't hesitate to stop me there. Vance, please, you too, Yeah. Okay, so let us go through this listing. We looked at FAM57B function, firstly, with regard to lipid metabolism, because of the claim that this was a ceramide synthase, this encoded a ceramide synthase. And in collaboration with colleagues at the Weizmann Institute in Tony Futterman's lab, we performed tissue culture assays with human cells, whereby we looked at the effect of FAM57B function on ceramide synthesis. And if you look at panel A here, you'll kind of get it. So on the y-axis is ceramide synthesis, and on the x-axis are the various constructs that were transfected into the cells. If you just put in control DNA or FAM57B alone, there is no ceramide synthase. If you put in the CER2, which is a ceramide synthase, you now see there is some ceramide synthesis. But if you mix the CER-S2 and FAM57B, you now see a great synergy of and an increase in ceramide synthesis. And so this tells you that there's some kind of modulating role of FAM57B as a ceramide synthase modulator. And if you follow on the other side of the slide, you can see for CER-S6, another ceramide synthase, co-transfection with FAM57B actually depresses ceramide synthesis. And so these data newly show that FAM57B is not a ceramide synthase per se, but can regulate ceramide synthesis. So that was very interesting. We were particularly interested in what cells from people affected with 16p deletion syndrome looked like, and whether or not they would show a difference in lipid profile relative to unaffected people, as might be predicted if FAM57B was playing some role in lipid metabolism in affected people. And so we cultured 16p deletion iPS neurons from the Simons Foundation, and we showed that, so this is a huge effort, but using these iPS-derived human neurons, we were able to show that there is an altered lipid profile in neurons derived from affected people relative to unaffected sibling controls. And here is a lipid, there's huge amounts of data here, but here is something that will summarize it for you. Here are lipid classes where concentrations increased and lipid classes where concentrations decreased in the 16p deletion neurons relative to control. And you can see that there are significant differences in the lipid cohort of these cells. And you can see, if you look at my diagram on the right, that some of these map to the ceramide pathway, not all of them, but some of them map to the ceramide synthesis pathway. So that gave us some great confidence and intrigue that there are changes in the lipid cohort in cells from affected people. And that's extremely important to make that connection between the predictions and the human cells, in my opinion. We then went back to zebrafish as our model, and we built gene-edited FAM57B zebrafish mutants. and looked at the lipid cohorts from brains isolated from these fish. And you can see, if you just glance here at the green and the purple dots, those tell you that there are differences. And there are differences both in the mutant fish, where both copies of FAM57B are knocked out, or in fact, in zebrafish, there are 2 FAM57B genes, but don't worry about that. And here also in heterozygotes, where there is just 1 copy of the gene knocked out. In the 16p syndrome, there is just 1 copy of the interval that is lost. So you are haploinsufficient. You have 1 regular chromosome 16 and 1 chromosome 16 that's lost, the 16p 11.2 interval. Okay, so if you want to be really rigorous, you can look at heterozygotes rather than mutants that have lost both copies. But in actual fact, you see changes in both situations. And what we could conclude is that the changes in lipid classes are overlapping, but not the same as in 16p deletion neurons, likely because other 16p 11.2 genes contribute to lipid metabolism in human cells also. Okay, but this gave us a sense that there was something that was changed with FAM57B modulation. The data, when we looked at the lipids that were affected, the data suggested that actually one of the things that would be affected would be the plasma membrane. And here is a diagram of a wild type lipid membrane and a predicted FAM57B mutant lipid membrane. Now, what I didn't know and what you might not know is that the double membrane, or the lipid bilayer of plasma membranes, has got different lipids in the outer leaflet, as well as in the inner leaflet. Did you know that? I think that's absolutely fascinating. So here, this is the outer leaflet, and the different color circles are particular kinds of lipids. And on the inner surface, you can see there are different lipids. Like, wow. And this is important in terms of proteins that can associate with these lipids. If you look at the predictions from the FAM57B mutants and the lipids that are changed, you'll see that the predicted structure of the plasma membrane is really different. Danielle went on to look and see if that was true by using a number of stains, including a cholera toxin B stain. And if you look at these panels, you can see here AB, a wild type fish, and in FAM57B mutants, you can see that there is a big increase or significant increase in these green dots, which are lipid rafts that are associated with various particular lipids in the plasma membrane. So you can see structurally, there is something different between the control and the FAM57B knockout plasma membrane. So that was very interesting. We went on then to ask the question of what the significance of these membrane changes was. And Danielle asked the question of, do proteins associated with the plasma membrane change between wild type and FAM57B mutants, particularly those that are found at the synapse? And she did this by developing a biotinylation mass spec assay where you biotinylate the plasma membrane proteins, pull them down, and then run them through mass spec. And she found that there were significant changes between the mutant FAM57Bs and the control AB fish. And you can see again, the green and the purple dots. Actually, you can see there's lots of dots here, but we've pulled out certain green and certain purple dots. The protein that was most depressed at the synapse, associated, not at the synapse at this point, but so the most depressed associated with the membrane is a protein called synaptotagmin. And you may know synaptotagmin, those of you who are neurobiologists. This is a key presynaptic regulator, and its association with membranes in this assay was severely depressed. So it's really fascinating. And it was fascinating when Danielle looked in zebrafish because she could see that overall, it's very faint here, but I can see it on my screen. There's actually no overall change in the level of synaptotagmin protein. But if you do an immunostain of zebrafish brains, you see that there is a real problem here. Here's a zebrafish brain, and the blue here is synaptotagmin, which is localized to the presynaptic area in various neurons. If you look in a mutant brain, brain, you can see there is synaptotagmin all over the place, like just in wrong places. It looks like a mess. And so we think that the synaptotagmin is no longer captured in the presynaptic complex, but is rather distributed along the axons and, of the neurons, or just inappropriately, so that it is no longer in its place where it is functional. And so this was really fascinating. Danielle went on to further examine synaptic composition by isolating synaptosomes, which is a way of pulling out the actual presynapse and postsynapse as a complex. And then she did a mass spec on these and looked to see what the proteins associated were in the mutant FAM57B and the ABs. And again, there are big changes, and you can find synaptotagmin is one of those changes. So she did the same thing in human cells that we gene edited to remove FAM57B, and I won't show you all the data, but there is real concordance that in human cells where this the ceramide synthase modulator has been knocked out, there are changes in synaptosome associated proteins. So this told us that there are changes in the lipid cohort, membrane structure, synapse associated proteins. And Danielle next asked about the significance of this with regard to brain activity. And she had previously developed, in conjunction with a company, Axion, a way to do multi-electrode array recordings on zebrafish brains. So here's the multi-electrode array, and here's the little zebrafish larva. And you can actually figure out which electrodes it's in contact with. Okay, so you can get several electrodes that are in contact with the brain and the spinal cord and do recordings and so on. So we have a lot of quantitative data, but the data I actually wanted you just to look at is this in the red box here, which I think is fascinating. Here is the brain tracing from wild type, and you can see there's lots of activity. Every peak is a brain activity. If you look at FAM57B mutant brains, it's kind of flat. It really is. And by any measure, you can see that there is a massive decrease in brain activity, and these things called bursts, where there are multiple neurons firing at the same time, were so depressed we had to change the scale in order to actually pull any out. So there is a massive association of change in brain activity with the knockout of FAM57B, suggesting that the changes in proteins and synaptotagmin, for example, and other synaptic proteins, may contribute to this change in brain activity. But of course, we would also like to know the significance for behavior, because the output of mental health disorders, neurodevelopmental disorders, is often changes in behavior, and that is what is so debilitating about these disorders. So we use the zebrafish, which is again a fantastic model to look at behavior. Danielle developed a doc startle assay where you have, let's see if I can, where here are the fish in their little wells. This is just a close-up of a multi-well plate. And you can see the fish move around and you can quantify them. We have a very nice apparatus in the lab here at Northeastern that quantifies movement and gives us the traces. And what we found is that when the light is turned off, the fish startle. That's what these peaks are. And it's repeated, and there's some habituation, but it can persist for a long time. But the FAM57B mutant fish were much lower in their responsiveness. You can see they're the purple peak here. They respond, but much lower, to a much lower level than the wild type fish. And we have some other behavioral assays that support this type of analysis. Okay. So what I have shown you is that we've been able to link FAM57B function with changes in lipid metabolism. And associated with that, of course, we don't know the exact causal connections at all, but associated with FAM57B disruption in both human cells and in zebrafish model are changes in synaptic protein association, synaptic activity and brain activity, and in behavior. And, you know, if we really extrapolate out, and clearly there is a great deal more work to do, we can suggest that an altered lipid cohort may contribute to 16p deletion syndrome symptoms, and that modified lipids may be targets for novel therapies. So how are we doing for time, Vance? Because I now did something that was what I was going to tell you, but then in honor of you as you know, a close group of colleagues, and because Craig hasn't seen these data also, I have a few more slides. How are we doing for time, Vance? I'm not quite sure.
You've got close to 15 minutes left, so you got plenty of
Okay,
time.
well, we certainly will not take 15 minutes because we need discussion, but let me zip through a few more slides that get to the question of other symptoms associated with mental health disorders, and the notion that there is a brain-body axis that is often co-opted or part of the landscape of brain disorders and mental health disorders. And I'm going to go on in just a few slides to share data. I've never actually presented this before because it isn't, yeah, I haven't presented it before, but it's in preparation for a paper in preparation that will tell you that not only are there brain symptoms with FAM57B knockout as our kind of indicator gene for mental health, this particular neurodevelopmental mental health disorder, but there is a body size aspect, and it's fascinating to try to understand the connections. Jasmine McCammon previously showed that in FAM57B heterozygote fish, there is an increase in body size. Monica Saxena went on to follow this up, and these are fish at, I think these are 7-day fish. No, they're probably actually 14-day fish. But anyway, that what you can see is here's a wild type fish. Here's a mutant fish with both copies of FAM57B knocked out. And here is a heterozygote fish. And if it looks bigger, it is bigger. And the quantification is down here. These fish are bigger in a number of parameters. What Monica went on to do was to ask the question of whether the bigger fish ate more. And she fed them with fluorescently stained paramecia, which the fish larvae love to chase and eat. And she could quantify by fluorescence how much they were eating over time. And you can see here that the wild type fish and the FAM57 double heterozygote fish do not have differences in how much they're eating. Okay, in fact, the heads look like they're eating a little bit less, but it's not significant. So this is not because they're eating more. There's something else going on. And we can hold that one because we don't know what it is. But a couple of things we do know that have to do with, you know, perceptions and how wrong you can be. FAM57B is primarily expressed in the brain. That's true in mammals, it's true in humans, it's true in mice, and it's true in zebrafish. And you can see this by these, you know, RNA-seq assays where you can see that the brain expression of FAM57B, look at the scale, it breaks here, is way, way higher than in skeletal muscle or adipose tissue, okay? Now, for various reasons, Monica also looked at a very important regulator of a lot of things, a gene called PPAR gamma, which is a major regulator of multiple processes and also part of the steroid hormone receptor regulatory family. And what she found was fascinating in that there is a reciprocal quantitative expression of PPAR gamma. So it is very low in the brain, but higher in muscle and very high in adipose tissue, where it's a key adipogenic regulator. So its expression was reciprocal to FAM57B. Furthermore, if you look in FAM57B, heterozygotes that have lost just one, or in this case, 2 copies of the gene, you now see that there is a massive change in PPAR gamma. This is all RNA expression, that all of a sudden PPAR gamma expression increases in the brain, increases in muscle, increases in adipose tissue. So as FAM57B expression decreases, PPAR gamma expression increases. And the fact that FAM57B is not normally expressed at very high levels outside the brain it doesn't mean that it's not acting outside the brain, because in these heterozygotes, this is what's changed. It's this low levels of expression in muscle and adipose tissue, and it's leading to changes in PPAR gamma expression. Interestingly, and I think this is very important, in the complete loss of function of FAM57B, you do not see these same changes. There is a change in PPAR gamma expression, but it's not this reciprocal change. It's as though you need some FAM57B and some some PPAR gamma to get this kind of inverse correlation. Okay. Where did we go with this? A number of places. One of the things that we were intrigued about was this notion that, unlike our hypothesis, FAM57B was not acting from the brain to control body size. I had thought it was acting from the brain. This was a control center. It was modulating neuropeptide hormones, leptins and things, and so controlling body size. But I don't think that's true, because we could see these changes in these other tissues. And so Monica did this great experiment, this explant experiment, something you can do so well in zebrafish. She explanted the brain and the pre-adipogenic region, and she cultured these in the presence of modulators of PPAR gamma. And so resiglitazone is an activator of PPAR gamma, and this GW9662 is an inhibitor of PPAR gamma. And so she took out these two regions, cultured them in the presence of these modulators of PPAR gamma, and then she looked at FAM57B expression. Okay, in this, so now isolated from the rest of the body. And here, the top lines here are the adipogenic region. Here's the control. This is what FAM57B expression looked like. These are the two FAM57B genes. And you can see when you add this inhibitor of PPAR gamma, the GW9662, now there's a big increase in FAM57B expression, indicating that in the isolated adipogenic region, PPAR gamma is keeping FAM57B expression off. Okay, and you can see here what happens to PPAR gamma. And in the brain, you see the other way around, where resiglitazone, which activates PPAR gamma, now inhibits FAM57B expression and does so in a tissue autonomous way. So I think this is very interesting and shows, that a hypothesis that a gene that is very highly expressed in one region is only acting from that region can really be not correct, which I think is true in this case. Monica went on, and I'm not going to show you all the data because it goes on for pages, to do RNA-seq from skeletal muscle, the adipogenic region, and brain to ask what changes in RNA gene expression we could see in FAM57B heterozygotes or mutants. And we could see a bunch. Now, what's fascinating, and you don't quite get it from these Venn diagrams, but they're in a way easier to look at, is that there are many changes in gene expression with FAM57B change. And remember, FAM57B is not a transcription factor, is a modulator of ceramide synthesis. So this is an indirect kind of effect, maybe through PPAR gamma, maybe through something else. But if you look, there are changes in adipose tissue, brain, and muscle. Some of the genes are upregulated, some are down. And what is fascinating is that although there is overlap in changes between these tissues, there are many, many genes that are specific, upregulated or downregulated specifically in adipose tissue, muscle, or brain. And if we looked, you don't get it from this, in the mutants versus the heterozygotes, there again are genes that are commonly changed between all of these, gene expression commonly changed between all of these tissues, but also differences. But if we were to superimpose the two, the changes in gene expression in the mutant fish, where we've totally knocked out FAM57B, versus the heterozygote fish are really different. They are almost not overlapping. So this is fascinating to me because 16p deletion syndrome is a gene dosage syndrome. It is a haploinsufficient syndrome. If you have one copy of FAM57B removed, or in our zebrafish model equivalent, you get a certain phenotype. If you have both copies removed, you don't just get a worse phenotype the same. You get a totally different phenotype. And you can see that at the gene expression level. And I think that is really important for these dosage regulated human syndromes, which copy number variant syndromes all are. They are usually haploinsufficient syndromes, and the output one would predict would be different than if you'd actually lost both copies. Often that is lethal, but certainly there is some kind of dosage effect here, and this, we can see that at the RNA-seq level. Okay, so this is the working graphical abstract, that there is some kind of mutually inhibitory relationship between FAM57B and PPAR gamma. And since, we don't exactly know how FAM57B would modulate PPAR gamma, but there's some sense of the intervening changes in transcriptional regulatory machinery that could be affected. I've shown you also that there is reciprocal levels of expression of FAM and PPAR gamma across brain, muscle, and adipose tissue. And that, if you knock out FAM57 or PPAR gamma, you get changes in the transcriptome. To circle back, I started this, a little part of the talk, with the notion that there was a body size phenotype associated with the heterozygotes in FAM57B knockout. And so here we have now a kind of brain-body connectivity with a single gene that is clearly not the only contributor to 16p deletion syndrome, but maybe one of the key regulators of the syndrome, that is not only connecting to brain phenotypes, but that is connecting to body phenotypes associated with the syndrome. So that's the data I have for you. And I want to acknowledge, the people who brought the data. Danielle Tomasello did all of the brain with collaborators, did the work on the lipid cohort and the brain activity assays. Monica Saxena was responsible for the work that I just told you about on the body connection. Here at Northeastern, the people in red. Adriana Van Duren, I would say, is not in my research group, but I am thrilled. She is a graduate student in Tova Day's lab. Sarah Barnes is a new postdoc in my group who has taken over this project. Cass Braziller is our animal manager, and Ashwin Lokapali is another postdoc in the group. And I'm thrilled to have my first co-op, Courtney Cagle, who is wonderful. And then there's a lot of other collaborators, including our collaborators at the Weizmann Institute, who have been so fantastic as helping us understand the function of FAM57B. So that is what I have for you. And I can put this other thing up here. Craig, I don't know if this is useful, but it's kind of perhaps something for us to be thinking about at Northeastern as we put together, the work, continue with our own research, but put together collaborations that cross animal models and cross techniques that, for any mental health disorder, there are many risk genes. And the notion is that each have a small effect. GWAS hits maybe give you sometimes a 1% increase in probability of being associated with a disorder. They're very small. That there are multiple metabolic enzymes that are part of these risk genes, that these may change the metabolome, and that these indeed are targets for small molecule screens that could correct the cell biology and the behavior associated and improve the lives of people who are affected. So that is what I have for you. I'm going to stop sharing.
Wonderful.
Oops.
Thank you, Hazel. That was fascinating.
Stop share. Okay, there. I stopped. Thanks very much.
We have some time, it's 12, but if you're still around and have a question or have any discussion or comments you'd like to make, now's your chance.
Yeah, this is Art Kramer. I'd like to ask a question to either Hazel or Craig. Thanks for the presentations. I enjoyed them both. I'd recently, in thinking about what you both had to say in terms of metabolic regulators and so forth, I was just reading over a recent paper in Science, February 11th, on caloric restriction in humans reveals immunometabolic regulators of health span. So I was wondering what the two of you thought about the CR literature and where it might be going and how it interacts with the kinds of issues that you were talking about.
Well, actually, if I may, Art, the CR literature goes back probably what, 40 or 50 years.
Sure, but it's really ramped up quite a bit in recent years.
You know, it's better not to eat than eat, kind of thing. It, is my general take on it. And listen, I haven't been a part of this for years in thinking about it, but I think it's, yes, that kind of fits. Maybe we should be looking at this also from by looking at biomarkers of metabolism, but looking at general metabolism, literally CR2, O2 utilization, so forth and so on, as other, indices. Yeah, you can just run it. You can run it just so hot, if I can use the metaphor, okay, in terms of the brain and feeding it. And the more you put in, the more, the hotter it gets, so to speak. And maybe that's a way of approaching this. So that was a science paper just recently, Art?
Yeah, it's the first author is SPADARO. It was February 11th. And, they talked about many of the issues that you and Hazel talked about in terms of metabolomics, of course, epigenetics. There's a particular gene that CR seems to downregulate, and that seems to be useful in decreasing inflammation. And as we know, inflammation is one of the hallmarks of aging, which is not a good one.
All right, thanks.
I think it's very interesting. I think the question of neurodevelopmental disorders versus disorders that arise with age, I think it's fascinating, right? Is there a fundamental kind of difference there? That I think so. I think so. And would caloric restriction have different effects on these different populations? We could certainly model this type of thing. It probably would do something. I can't imagine, that if we starved our zebrafish, there would not be some profound change in aspects of their metabolism and behavior,
Well, I think the notion is that CR isn't starvation. It's a balanced diet. It's just much less than we eat, especially us Americans, which are terrible at eating too much.
Totally, Art. Totally understand exactly. So yeah, I think very interesting.
Thanks.
Yeah, just to follow up on Art's question, since he asked kind of a general question, I have kind of a general question also. I'm an engineer and I look at the biomechanics of the brain. So you can probably guess what my question would be. What might be the role of exercise and motion and movement? Maybe to put it in that, I know that's a broad question, probably difficult to answer, but let's just say something a little tighter maybe. If I have those zebrafish swim a little bit extra 20 minutes a day on their swimming treadmill, or however you want to do it, and how would that impact the results? And is it too complicated to say?
Looking at Sarah Bonds, who I see on the in the meeting here. What do you think, Sarah? We could do it. Thanks for the question, Francis. It's a great question. Craig could certainly do it with his rodent models. I would be amazed if it doesn't do something. I just think diet, exercise, these things are so profound. You and I know if we actually have our, 10,000 steps a day, it feels different. And it's not, psychological is like a garbage term. It feels different because there's a change in physiology and biochemistry. So I'm totally, the question, and this is for, Art, I'm looking at you, you're the CBH group, and for our fantastic movement biologists here at Northeastern. These are really great questions that could be interfaced, I think, with people, even at this time,
Yeah, I think Hazel, I think exercise.
Do you want to take that one?
Hazel, I think exercise is an easy one because as Craig said, there's been work on for decades on CR. There's been work for decades on exercise, all the way down to the mitochondria, the telomere length, nerve growth factors, synaptogenesis, angiogenesis, neurogenesis. In fact, when I was a young assistant professor, I worked with a guy by the name of Bill Greeno, who's since passed away, certainly one of the luminaries in brain plasticity for many decades. And that's what Bill focused on. I know that Craig is focused on exercise. We know a lot about the molecular and cellular biology of exercise. If you're interested in the brain, of course, we know a lot about the biomechanics. And for the last almost 40 years, I've been studying exercise effects on human brain and cognition using MRI, FNIRS, EEG, you name it, we've used it, we still use it. When I came to the university in 2016, my friend Joseph was kind enough to give me a $5 million check. So we bought a complementary machine to that Craig has. We have a 3-Tesla MRI system in the basement of ISEC that we can study non-invasively or relatively non-invasively human brain changes in structure function as a function of exercise. And, you know, for the most part, exercise effects are much larger if the organism is young. But interestingly, you know, maybe it was 20 years ago, we thought that aging was a one-way street. And as we get older, there's no hope. Don't even bother. Sit in your rocking chair and wait for the end. But we know from the exercise literature that's clearly not true, because we've even seen neurogenesis in the dentate gyrus of the hippocampus in older animals. And we see changes in connectivity using Granger causality and other techniques with human data. So exercise, diet, sleep, reduction in stress, all of these lifestyle variables are critical. We know a lot less about the microbiome because that's a relatively new field, but you know, that's clearly a way to go.
So you know, Art, I think this is just to jump in. I think it's you know, it's very interesting. What we don't know really is for specific syndromes. So you know, we know sort of across populations, but to target specific syndromes, that seems to be the kind of key thing here. If we pick out, you know, in our case, we've focused on 16p deletion syndrome at the moment. What would exercise do for that particular population of affected people? And that, I think, is where there is still work to do. And so I think, Francis, your question is a very good one, because there is kind of the vast amount of data, and then there's the targeted data to particular disorders and to particular affected groups. And I do think that there is a space and landscape to move further in that direction.
Hey, Hazel, I think you're exactly on target. And certainly we don't know a lot about a lot of disorders, but we do know about exercise effects on multiple sclerosis. It actually enhances white matter integrity, which is pretty amazing because that's a white matter disease. Alzheimer's, Parkinson's, ADHD, autism. So there are a number of populations that have been studied, not with particular deletions. Of course, with animals, it's easier to do knock-ins and knockouts. We can't do that with humans, not ethically anyway. So I think to target some of these disorders where we might be able to benefit various systems. Parkinson's is an easy one in a sense, because the basal ganglia gets depleted of dopamine, and one of the effects of exercise is to increase dopamine uptake in the basal ganglia. So I think exercise, diet, and all these other things need to be targeted to particular groups. I couldn't agree more.
Just a further comment on that. Thanks for that answer, by the way. So my group has been really interested in looking at the natural motion of the brain during the cardiac cycle. And I've been studying a disease for a number of years called Chiari malformation. And we looked at it particularly for that disease. But we came across my collaborator at Emory told me about a technique called DENSE that is typically used for the heart, but they've started using it for the brain, where you can measure really small motions quite accurately. And as an engineer, we developed the apparatus to measure just how accurate this was. We took it down to Atlanta, and you can measure somewhere in the neighborhood of 20 to 30 micron peak motion during the cardiac cycle. So we looked at it for our patients, for our Chiari patients, and we noticed that their brain tissue, cerebellum, brainstem were moving about twice that of a healthy volunteer. But maybe more interesting for this group is that the healthy volunteers, there was quite a wide variation. In other words, you have a healthy control that has maybe 80 or 90 microns of motion, and another healthy control would have 200 or 250 microns of motion. How is that important for mental health, and how does that go towards Alzheimer's? I don't know, but I think it's another parameter to check. It's kind of a lot of work to check, right? Because you have to do the MRI scans, and you have to have a hypothesis as to why something moving around would play a role in the complex system. But I would like to start doing those brain motion studies in the MRI scanner at ISIC soon. I just joined the faculty this year, so I'm very interested to start working on that at the new 3T scanner in ISIC.
Francis, we can do those studies. In fact, we need some hardware, but not a whole lot, and some software. In terms of MRI, it's called magnetic resonance elastography. And it's a very bizarre kind of system, but it essentially is a system that fits inside the head coil if you're interested in brain. And you will actually vibrate the head at a particular frequency and particular phase. And what it allows you to do at a fairly gross level compared to the resolution you're talking about is to look at the elasticity, the mechanical properties of tissue, be it gray or white, and of course it depends on where it is. And the interesting thing about MRE, one of the developers is Brad Sutton at the University of Illinois. Another one is Curtis Johnson at the University of Delaware, who we will target for a hire next year if we're fortunate. I'd like to get Curtis here. And interestingly, we and others have looked at these parameters, and the mechanical properties of tissue tend to be more sensitive to brain changes, structural changes, than volume and other anatomical measures in the hippocampus as a function of exercise. So they're certainly complementary to other things we can measure in humans and in animals, of course. So I think it's very possible. We need a little bit of hardware, a little bit of software. Money is only money. It's just a matter of writing a grant to get it right, kind of a small MRE. Craig might even have this device in his magnet. I don't know.
Well,
No, we don't. No, we don't. But yeah, the MRE is exciting. Actually, so question to you, Francis. Number one, is there a rat model of this condition that you study?
The short answer is no, but we do get to a rat model if we create syringomyelia, which is very often associated with Chiari. So about 40% of Chiarians get syringomyelia. That's a cyst inside their spinal cord. And we have a collaborator in Ohio that has figured out how to inject some, I can't remember what they inject. They inject something into the rat, and then it causes an injury, and then a syrinx forms below it. So we can't do Chiari malformation per se, but you can study syringomyelia, which is another important issue. And syringomyelia, we can have that disease can come from injuries, car accidents, and military injuries as well. So it's still an important injury. So,
One thing, I have a lot of questions. I don't know whether we want to do this offline or not.
Yeah.
I'm sorry. One thing I can add is that we can implant a balloon at the foramen magnum level, inflate the balloon, and the balloon will simulate Chiari, because Chiari, you have the cerebellar tonsil going down through the foramen magnum and affecting the fluid flow. So we can simulate, but it's absolutely right, as Dr. Loss said, short answer is no, but we can simulate. I don't know if that makes sense.
Well, I would be very interested to continue this conversation. Maybe I've already taken up a lot of the time on this group.
I put a question in the chat, Francis.
So is CSF volume and movement change related? The short answer to that is yes, because Chiarians have a lower cerebellar tonsil, so there's sort of less volume in the bottom half of the brain, sort of and those are the people that have more motion. How is it connected? We've been looking at that for a number of years, and as an engineer, you can imagine that's like I feel almost guilty that I can't explain why that motion is, but the motion is so subtle. We don't know if it's from the blood vessel, patterns inside, the complex, maybe their structure is a little different. And maybe it's a little bit, some researchers propose a Bernoulli effect, like you get this tight junction. I'm not sure if everyone's familiar with the Bernoulli effect, but you get a tighter junction, then the pressure drops. So you kind of suck the cerebellar tonsils down, but that doesn't entirely make sense either. So we want to put in a grant to try and understand that motion. And we want to do it in such a way that it looks at more diseases than just Chiari. We want to, my big question in Alzheimer's is brain motion the same in an Alzheimer's patient as someone who does not have Alzheimer's? I'd like to do that at some point, but yeah, there's very little, in short, there's very little known as to what's going on with that motion, but it definitely is related, because ones with a tighter junction, less space, they get more motion. So your intuition is correct.
Well, zebrafish is these models are great for CSF movement, Francis, as well as the.
Are they really? Zebrafish, I did not know.
Yes, we published on this.
Yes,
I passed the project on to my postdoc, who's now setting up her own lab. But yeah, it's a really, you can get there very accessible, so you can actually see things very accessibly.
These zebrafish do everything, huh?
Totally.
That's amazing. I did not realize that.
Any other comments, questions? I had more of a kind of curiosity one about the FAM57B missing zebrafish, which is it? So you mentioned body size was something that was noticeably different. Is there, and I think you said that those pictures were taken at like 14 day zebrafish. Was there, I guess I also don't know how long zebrafish live, but is there any sort of like long-term impact of this thing? Is 14 days long for a zebrafish, or?
Oh, 14 days is equivalent to a, I would say a middle schooler.
Okay.
So a young person. It's a juvenile.
Got it. When they reached adulthood, would they even out, or did the difference persist throughout life?
So they evened out, but we haven't looked at specific tissues to know whether specific tissues changed in size or in volume. The adults clearly have issues. They don't mate very well, for example. But we decided to pick a stage of development where there was a kind of very clear effect, and that's if you want to take the bold move of extrapolating to people who are affected, that is where the sort of greatest body size difference is seen. It's seen in young people, and then as time goes on, things can normalize a bit more.
Thank you. Okay, well, if we are low on questions, and I think we will probably call it here. Oh, Craig's got something though. Oh, you're muted.
I was saying goodbye.
Oh, goodbye. Wonderful. Thank you once again. Thank you, Craig, and thank you, Hazel, for your wonderful presentations. Thank you for everyone for being here and for contributing. There will be a recording available if you'd like access to it, let me know and I can give it to you so you can pass it on to other people who might be interested. And yeah, thank you once again, everybody.
Thank you. Let us know if you have an interest in collaborating or some ideas for how we can move forward here at Northeastern. Be lovely to hear from you or others that you think should be pointed in our direction. Hey, Craig?
Yes.
Awesome.
Thanks,
Bye-bye.
man.